Have you ever pondered upon the uniqueness of your identity? Ever wondered how DNA fingerprinting helps in identifying individuals? DNA fingerprinting, or DNA profiling, is a remarkable technique that allows us to determine the nucleotide sequence at specific parts of the DNA. These sequences are unique to each human being, akin to a biological barcode. This process was brought to light by Sir Alec Jeffrey at the University of Leicester in 1985. The DNA of every human being on the planet is 99.9% the same. It's the remaining 0.1%, roughly 3 million base pairs, that makes us unique. This includes what's often referred to as junk DNA. These non-coding sequences may not code for proteins, but they make up 95% of our genetic DNA, repeated many times over, and play a crucial role in our individual identities. As we delve deeper, we will uncover the fascinating principle behind DNA fingerprinting. To understand DNA fingerprinting, we must first grasp the concept of satellite DNA and its polymorphism. This DNA, although non-coding and often referred to as junk, is pivotal in defining our genetic individuality. It makes up a staggering 95% of our genetic DNA and can be separated from the bulk DNA during density gradient centrifugation, hence the name satellite DNA. Now, within this satellite DNA, we find sequences of bases repeated in tandem, earning them the name tandem repeats. Depending on their length, base composition, and number of repetitive units, these tandem repeats are categorized into subgroups like microsatellites and mini-satellites. Now, here's where it gets interesting. These satellite DNAs show polymorphism, a term we use when a variant at a locus is present, with a frequency of more than 1% in a population. These variations are the result of mutations, changes in the DNA sequence that occur over time. These mutations in the non-coding sequences accumulate and form what we know as DNA polymorphism, a variation at the genetic level. The junk DNA regions are thus composed of length polymorphisms, which show variations in the physical length of the DNA molecule. At specific loci on the chromosome, the number of tandem repeats varies between individuals. There will be a certain number of repeats for any specific loci on the chromosome. These differences in the number of tandem repeats, whether they're short tandem repeats with 2 to 5 base pair repeats or variable number of tandem repeats with 9 to 80 base pairs, contribute to our unique DNA profiles. It is important to remember that a child inherits 50% of their DNA from each parent. Therefore, the number of variable number tandem repeats at a specific area of the child's DNA may differ due to insertion, deletion, or mutation in the base pairs. These variations, known as length polymorphisms, form the basis of individual uniqueness in DNA fingerprinting. Now let's understand the significance of tandem repeats in our DNA. Tandem repeats are fascinating aspects of our genetic makeup. These are sequences of base pairs that occur adjacently in our DNA, repeating like a mantra of life, echoing through our genome. Now imagine these repeats as a series of patterns in a tapestry, with each pattern representing a specific tandem repeat. The size and number of these patterns can vary greatly among individuals, adding to the richness and complexity of the genetic tapestry. Now, there are two primary types of tandem repeats that we're going to focus on: short tandem repeats, or STRs, and variable number of tandem repeats, or VNTRs. STRs are like the smaller, intricate patterns in the tapestry, consisting of just 2 to 5 base pair repeats. VNTRs, on the other hand, are the larger, more prominent patterns containing repeats of 9 to 80 base pairs. Here's where things get really interesting. Every individual possesses a unique combination of these tandem repeats, particularly VNTRs. Think of it like the specific arrangement of patterns in a tapestry, with no two tapestries being exactly alike. This is due to the fact that the number of VNTRs at a particular area of the DNA can change due to insertion, deletion, or mutation in the base pairs. This distinctive combination of VNTRs is passed down from parents to offspring, with the child receiving half of its DNA from each parent. Consequently, the child's DNA will possess its own unique arrangement of VNTRs, adding another layer of complexity to the genetic tapestry. In essence, these unique VNTR compositions form the basis of DNA fingerprinting. They allow scientists to distinguish one individual from another, much like how a keen-eyed art enthusiast can distinguish one tapestry from another based on the arrangement of patterns. So, the next time you marvel at the uniqueness of your own identity, remember the tandem repeats in your DNA, those intricate patterns in your own genetic tapestry that contribute to who you are. These distinctive VNTR compositions lie at the heart of DNA fingerprinting. Now that we understand the principle, let's dive into the process of DNA fingerprinting. The journey begins with a sample of an individual's DNA, which could come from blood, skin, hair, or other biological materials. This DNA is then subjected to the action of certain proteins known as restriction enzymes. These molecular scissors have a unique ability. They can cut DNA molecules at specific sequences known as restriction sites. Imagine a long, winding highway with toll booths placed at specific intervals. Similarly, the restriction enzymes navigate through the DNA highway, stopping at these toll booths or restriction sites to make their incisions. But the placement of these toll booths isn't the same for everyone. A single change in nucleotide may create new toll booths or abolish existing ones. This is where the magic happens. When the DNA is digested with a restriction enzyme, it results in fragments of different sizes. These fragments are like puzzle pieces, each unique in size and shape. The distribution and arrangement of these pieces form the so-called fragment pattern. But how do we see these invisible puzzle pieces? Enter gel electrophoresis. This technique separates DNA fragments based on size, creating a visual representation of the fragment pattern. The smaller fragments travel further, while the larger ones lag behind, much like athletes in a race. The result is a distinctive pattern of bands, similar to a barcode. The next step is to transfer this pattern onto a nylon membrane, a process known as southern blotting. This membrane is then exposed to a radioactive probe that binds to the DNA fragments, creating a radiographic image or autoradiogram. This autoradiogram, or DNA fingerprint, is unique for each individual, save for identical twins. It's like a molecular signature, one that remains unchanged throughout a person's life. It can be used to establish paternity, solve crimes, and even trace human migration patterns. DNA fingerprinting thus offers an accurate and reliable method for individual identification owing to the unique patterns of our DNA.